So when you talk of when we talk of surface molecule, cell surface protein, all right, the common designation you see in the literature is what? CD. CD, right? And what does CD mean? It means cluster of differentiation. So the molecules, these are molecules present on typically, usually white blood cells, right? White blood cells. As you can see, most of the cells that are talking about here are white blood cells, right? Most of the cells that are talking about here are white blood cells. That's T lymphocytes, B lymphocytes, natural killer cells, macrophages, besides hematopoietic stem cells. These ones are in the bone marrow there, but most of these are white blood cells. So all of these are, you see that denoted by CD, CD, CD. What does that mean? It means cluster of differentiation. Okay. So when you identify a certain CD on a white blood cell, so this belongs to a T cell, this one belongs to a B cell, this white blood cell belongs to a B cell, this one is a macrophage, this one is a natural killer cell or bone marrow stem cell. Very important for you to know. Why is that? Because, like I said, two main reasons. One of the main reasons is tumors. So when you have a tumor from white blood cell, either leukemias, lymphomas, you use these CDs to identify the origin of that lymphoma. Is it coming from a T cell? So you say T cell lymphoma. Is it a B cell? You say it's from B cell. Is it macrophage? You say it's a tumor from macrophage, and so on and so forth. The other thing is infections. Why is that so? Some of these markers you're seeing here, right? Some of the markers you're seeing here are very important for, uh, are useful as cell surface receptors that infectious pathogens will use to infect these white blood cells. The most common here is this one, CXCR for CCR5, all right? So this is a cofactor that HIV uses to infect CD4 cells. Now, HIV affects two types of cells, T cell and macrophages, right? It infects two types of cells, T cell and macrophages, and it uses this CXCR4 and CCR5 to infect the macrophage. So it will use CXCR4 for the CD4 T cell and to use CCR5 here for the macrophages. Very, very important. All right. So that's one of the main. Then you see here Epstein-Barr virus uses CD21 to invade what? B cells. To invade B cells. Right. So very important there. Like I've said earlier, and the third reason, of course, which is the one I've mentioned, to help you identify the type of white blood cell. So it's very important for you to know that when you see CD3, right, CD3, CD28, or CD3, CD4, CD8, you're talking about T cells. CD4 for CD4 helper T cell, CD8 for cytotoxic T cell, right? And then regulatory T cell have CD25 as well with CD4. Then for the B cell, right, you're talking about CD19, 20, 21, these three. Very important, right? To help you identify the that this is of a B cell. So they tell you, oh, do you have a tumor and it has CD19 on the surface, CD20, CD21, you know that they're talking about a B cell, right? Or they tell you they produce a drug that targets a cell with CD19, CD20, CD21. On that, you know they're talking of a drug that targets B cells. And then for the others, this one, NK cell, remember we talked about it before previously. We said CD16 is an FC receptor that binds to the FC portion of immunoglobulins. So NK cell have two main receptors, CD56 and CD16, right? And then whenever you hear, moving on to macrophages, whenever you hear a macrophage, CD14, they are talking about macrophage. CD14 is talking about macrophage, right? And this, if you remember, we mentioned toll-like receptor, right? This is actually CD14 on the cell of the macrophage. Look at it, the receptor for PAMP. This is pathogen-associated molecular patterns, which is used to recognize different structures of microbes like LPS, right? Lipopolysaccharide for bacteria, gram-negative bacteria. So TLR, toll-like receptor, also CD14 on the surface of the macrophage. Then whenever you hear CD34, think of bone marrow stem cell. All right, CD34, think of bone marrow stem cell. Okay, so moving on from that. So for let me just illustrate one more thing here before I move on from that. So look at this. So for your HIV on the surface has this CD21 and CD20. All right, CD21 and CD20. Yeah, I said CD21. Sorry, GP120 and GP41. So this is on the surface of the HIV. All right, and it has this structure. And what's it essential for? To bind to CD4 on the surface of CD4 helper cells. And then look at the chemokine receptor. That chemokine receptor said this is CXCR4. This. So this is what it needs to help it. You can see with that CXCR4 here, it allows the GP20, 120, and GP41 to be able to allow to for the HIV to enter inside the CD4 T cell. So that's why for the macrophage is CCR5. Okay, so moving on, we have the Anergy. So anytime you hear Anergy, is that the T cell is unresponsive. So this occurs with both B and T cell. So you have the both T and B and T cell become anergic. That's they will become unresponsive to an antigen that they have been exposed to. Typically, when the second signal, remember I said the co-stimulatory signal is between B7 on the APC and on the T cell. T cell is what? CD28. All right. Let's look at that signal again one more time to remind ourselves. Look at it here. So whenever this B7 or CD80, CD86 fails to bind with CD28 on the T cell, this T cell will now become anergic. So if this signal fails to happen, the T cell become anergic or unresponsive to this antigen here. All right. So that's what it means with Anergy. So typically happens when you don't the second or signal or co-stimulatory signal is aborted. And of course, it says another mechanism of self-tolerance. All right. So it's another mechanism of self-tolerance. So now the next thing here, talking about active and passive. This is also talking about with respect to vaccination as well. So there are some types of vaccines that are active. Some type of vaccines offer active immunity. When it's active immunity, you there's on exposure to the antigen, it will lead to activation of white blood cells. There are both B and T cells, for example, and those B and T cells will go in and become memory B cells after getting rid of the pathogen. So that's where they talk about memory, all right? And you can call in two main ways. You can have it either from natural infection or from vaccines. That's whether your toxoid or whatever. And for passive, this is where you have preformed antibodies. So from the mother to the fetus, particularly IgG, right? IgG going from the mother to the fetus to protect the fetus or the baby against pathogens, all right, based on what the mother has been exposed to. So you talk of passive, that's preformed antibodies. And this one, the onset is rapid because the antibodies are rapidly immobilized. So, but the problem with it is short duration, while for active, longer duration because you're stimulating B and T cells, and when they are done, they will become memory B cells or memory cells. So examples of this, you have them in here, breast milk, humanized monoclonal antibodies, IgG crossing the placental, for example. So you clinically speaking, you can prepare passive immunoglobulins, give it to patients. So for some disease conditions like rabies, for example, you have a combination of both active and passive immunity, active and passive immunization, right? To protect the individual against. So there are two main that combine those two main disorders where they combine active and passive. That's rabies and hepatitis B. So you're giving them both a vaccine and you're giving them IV immunoglobulins. So you're offering them the IV immunoglobulin will act rapidly to protect, to neutralize the virus, while the active form of the vaccine will work to generate long-term memory. memory white blood cells, right? So whenever you hear of combination of both active and passive, think of hepatitis B and rabies infection.